Okay, so welcome to this next video in which we're discussing the effect of sympathetic stimulation on the heart. Okay, so we now want to discuss how we get these cyclic AMP oscillations. Right, so for this we need to discuss calcium inhibition of the adenylyl cyclase 5 and adenylyl cyclase 6 enzymes. So let's draw our adenylyl cyclase 5 and adenylyl cyclase 6 enzyme. So here's this first transmembrane domain here, and then you have this second transmembrane region here, the TM2 region, with these 6 membrane-spanning alpha helices. And basically, when the enzyme is in its active state, remember the C2A domain is dimerized with the C1A domain there. So here is the C2A domain here, so I'll highlight that in blue. So in blue here, this is the C2A domain. And then the C1A domain is in red here. So they dimerize together and form this active enzyme. However, at the center of this enzyme, there is a magnesium ion. Okay, so from the cytoplasm, when these two regions dimerize, so when C2A dimerizes with C1A, then a magnesium ion from the cytoplasm is going to come in and sit at the center of this enzyme. So I'll put it here. Okay, so this is a magnesium ion at the center here. Right, now, this magnesium ion is really important for the ATP molecule to come in. So remember, the way this enzyme works is an ATP molecule has to come in, it has to bind in there, and then what you have to do is cut the ATP up. So let's just have a reminder of the reaction that the adenylyl cyclase 5/6 is catalyzing. So this is an AC5 or 6 that we're talking about here. So here is our ATP molecule. So here are these 3 phosphate groups. Here's the 5th carbon. Here's our ribose sugar here. Okay, and here's our adenine organic base on here. So this is adenosine triphosphate. What's going to happen is this adenosine triphosphate is going to come in here. Now all of these phosphate groups here, the alpha, the beta, and the gamma phosphate groups of the adenosine triphosphate, they all have negative charges. So this is going to be coordinated by the magnesium ion, basically. Or in fact, the phosphate groups are going to bind to the magnesium ion that's at the center of this enzyme. And that's how you're going to get the ATP molecule to come in. Then what will happen is the enzyme will catalyze the cleaving of this bond here, so that you get this pyrophosphate molecule here, which remember you can abbreviate as PPI. Okay, so this is 2 phosphate groups bound together, the beta and the gamma phosphate of the ATP left behind, basically. And then what's left is you have an AMP, an adenosine monophosphate, but you do more than that. You convert it into cyclic adenosine monophosphate by binding this single phosphate group that's left to the 3rd carbon, like so. Okay, so here is your cyclic adenosine monophosphate. So this is cyclic AMP, CAMP. Okay, so let me colour these things in. So here is our organic base in green here, in light green. Here is our ribose sugar in blue here, which is this 5-membered ring where one of the members is an oxygen rather than a carbon, and then you have the 5th carbon of ribose stuck off at the top there. Okay, and here's the phosphate. So, what happens is the ATP comes in, the negative charges of the phosphate groups bind to the magnesium ion, and then the enzyme catalyzes the breaking of the bond between the alpha and the beta phosphate. So this is the alpha phosphate, this is the beta, and this is the gamma phosphate of ATP. That gives you the pyrophosphate, and then you bind the alpha phosphate to the 3rd carbon down here to create cyclic adenosine monophosphate. Okay, now once that has occurred, you then need to remove the cyclic AMP and the PPI from the active site. Now, that happens normally when you've got a magnesium bound there. But if calcium concentration goes very high in the cytoplasm, what do you think might happen? When the enzyme, well, what is going to happen basically is that calcium is going to displace the magnesium. So it's going to chuck that magnesium ion out, and in comes this calcium ion, which is slightly bigger than the magnesium ion. It's further down in the alkaline earth metals than magnesium. It's going to come in there, sit in there itself, and you might think that that's going to totally break the enzyme, but actually it doesn't. The ATP still comes in, okay, because, these phosphate groups can still bind to the calcium ion. It's got a divalent charge. They're quite happy. And then what happens is the proteins still catalyze the reaction where they break off the pyrophosphate and they produce cyclic AMP. The problem then is that these things don't leave the active site. Well, actually, cyclic AMP leaves the active site, so cyclic AMP goes. The problem is that the pyrophosphate doesn't. The pyrophosphate stays stuck in the active site with the calcium. And the reason for this, the reason that magnesium can get rid of the pyrophosphate and the calcium can't, is that magnesium is smaller. So let me try and explain this to you. Here is our magnesium, okay, and here is our calcium. Okay, and I should have drawn this further down. So, basically, when you bind things to these ions via electrostatic interactions, what you can do is, in the case of magnesium, you can put one thing here, you can put one thing here, you can put another thing here, and another thing here. Then, you can also put one thing above it, i.e. out of the page, and one thing below it, i.e. into the page. So it can coordinate 6 other ions, basically. And these bonds are known as coordinate bonds. So we say that the magnesium ion can coordinate 6 other chemical species. It has 6 coordinate bonds. Calcium is bigger. It can coordinate more things. What it can do instead is it can form 8 coordinate bonds, and that I won't be able to show this in the same way as that. So instead what I'll do is I'll draw a cube around the calcium ion, okay, and basically the corners of the cube represent where the chemical species that are being coordinated with the calcium ion will sit. So basically you'll have a species sitting at each one of these corners of the cube, and there's 8 corners of the cube. So you'll put one here, one here, one here, one here, that's 4. One there, one back there, another one here, and another one here, that's another 4. So that's 8. So, right. Basically, the way that you shove the cyclic AMP and the pyrophosphate out of the active site of the enzyme in the case where you have magnesium is that you bring in portions of the protein which interact with the magnesium ion. So you bring in 6 things which interact with the magnesium ion, and then there's no spare bonds for the pyrophosphate or the cyclic AMP, so they leave. Okay, whereas calcium has 8 coordinate bonds. So you bring in these 6 things again, you fill up 6 of the sockets, but there's still 2 left. So the cyclic AMP goes off, but look, pyrophosphate, 2 things. One sits here, one sits here. They've got those 2 extra sockets around the calcium ion that they can sit in. And when the pyrophosphate remains bound in the active site of that enzyme, it means that you can no longer actually bring in another ATP molecule. So yes, you made one cyclic AMP molecule, great, but the enzyme is now inhibited because the pyrophosphate is just stuck to the calcium ion at the center. So calcium inhibits adenylyl cyclase 5 and 6. So we know that we have calcium spikes inside the cytoplasm of these cardiomyocytes. So we know that calcium concentration in the cytoplasm of our cardiomyocyte versus time is oscillating. So it goes up, comes down, rests, comes up, goes down, rests. Now, if we stimulate our cell with a beta-1 adrenergic agonist, so for instance noradrenaline, then we are constantly trying to activate these adenylyl cyclase 5/6 enzymes, but they're only going to be successfully active when the calcium is low. So when calcium is high, you're not going to have the activation of the adenylyl cyclases, so you're not going to get the production of cyclic AMP, And then in between, when the calcium goes low, that's going to mean that the adenylyl cyclase 5/6 enzymes, they can start working now. They go up, they start producing cyclic AMP, and then when the calcium starts going up for the next time, that's when the cyclic AMP falls down, because the adenylyl cyclase 5/6 enzymes are turning back on, are being inhibited again by the calcium. So what you're going to get basically is out of phase oscillations of cyclic AMP in the cytoplasm of the cell. Okay, so you'll have cyclic AMP going up exactly when calcium is low, and when calcium is high, cyclic AMP will be low. That's what's known as out of phase. So they are said to be out of phase. Okay, or antiphasic would be another name for that. Antiphasic. Okay, right. Now, why is this important? Okay, it's a nice phenomenon, but this is actually important for the increase in inotropy that the cyclic AMP signaling is going to produce. Okay, so you're only going to get high cyclic AMP levels when calcium is low, i.e. in the middle. The cyclic AMP is going to activate the protein kinase A. Okay, so you're going to get the phosphorylation of the L-type voltage-gated calcium channels and the ryanodine receptors when calcium is low. And then when calcium goes high, i.e. when it's up here, the cyclic AMP goes low, so the phosphorylation of them stops. They'll have their phosphate groups removed by phosphatase enzymes. So what the brilliance of this is that you inactivate the L-type voltage-gated calcium channels and the ryanodine receptors. You take off those phosphate groups just as the calcium level gets really high and you need to start coming back down. Because if you kept those phosphate groups on, then what it would lead to basically is it would lead to those channels being continuously overactive. So you'd be continuously letting in too much calcium. So you'd have a basal sort of release of calcium, and what it would do is it would make it very difficult to keep a 100 nanomolar resting calcium concentration. Okay, so let me say this again. So, when cyclic AMP is high, protein kinase A is going to phosphorylate the L-type the L-type voltage-gated calcium channel and the ryanodine receptor type 2. So let me quickly draw these. So here's the L-type voltage-gated calcium channel on the plasma membrane, and here's the ryanodine receptor 2. Now, this phosphorylation increases their conductance to calcium even at rest, so they'll be releasing more calcium into the cytoplasm, both of them, at rest than they would do if they were not phosphorylated. Now, the genius of this is that you're going to only phosphorylate them right before you need them to become active, and then when you're in this down phase here, when you want them to be turned off, basically, that will be when calcium is very high. So calcium will be inhibiting the production of cyclic AMP, and therefore inhibiting the activation of the protein kinase A enzyme, and therefore inhibiting the activation of the L-type voltage-gated calcium channel and the type 2 ryanodine receptor. So you're going to have these two not being activated when you need the calcium to come back down. So what the brilliance of this cyclic AMP oscillation is that it gets these activated just when they need to be activated, i.e. it gets them activated when you're going on the upstroke of this calcium spike, and then they'll become inactivated in the downstroke, and they'll remain inactivated in this low point here, and then they'll become activated when you're just about to go on the upstroke of the next calcium spike, basically. So that's why the cyclic AMP oscillations are extremely important for the positive inotropic effect of these beta-1 adrenergic agonists. Okay, so we'll continue our discussion of the sympathetic nervous system's effect on the heart in the next video, where we'll discuss the chronotropic effects, i.e. the effects on the sinoatrial node.